Today we are going to talk about genome sequencing in humans and the different types of loss-of-function variants to answer the question, what can we expect if we get our genome sequenced? Our group number is A2 and is comprised of Haley Hunt, Malvi Johnny, Emma Jensen, Kelsey Roof, and Kara Strasser. Hopefully by the end of this discussion you will understand the results obtained, the implications of this experiment, and why further research is still needed.
Human genome sequencing is the mapping of all of the chemical building blocks that compose our DNA. This includes the DNA within each of the 23 human chromosomes and sex chromosomes. These chemical building blocks include nitrogen bases, adenine, guanine, cytosine, and thymine. The Human Genome Project was completed in 2003 and identified over 20,000 genes within the human DNA. These sequences used in the project were compiled from a variety of sources and do not represent a single individual's genome, but rather applies to all humans. This is possible because we all share the same basic set of genes. DNA sequencing is completed using a variety of steps in which some chromosomes are broken into smaller units and analyzed. The shorter fragments are used as a template to make more pieces, which are separated by gel electrophoresis. The bases are added to the sequence and are evaluated, concluding the DNA sequencing. Each human genome contains variation from one individual to the next. Loss-of-function variants represent changes within the genome that alter a protein's function. The recent experiment, a systematic survey of loss-of-function variants, was a substantial sequencing project that altered the way we perceive these variations.
The point of this experiment was to determine not only the prevalence, but also the difference between disease-causing loss-of-function variants and those that seem to have no effect on human fitness. In order to represent the human population as a whole, the 185 genomes that were used in this experiment included individuals from Nigeria, China, and Japan, and individuals of Western European origin. Stringent filters were applied to the 2,951 putative loss-of-function variants from these 185 genomes in order to account for any false positives and to find high-confidence loss-of-function variants.
Loss of function, also known as LOF variant, is a mutation within one's chromosomes. Its results are reduced or eliminated protein functions, which support three different hypotheses: less is more, and less is nothing. The less is less hypothesis occurs when the single mutation causes gene loss to be harmful, which results in diseases and disorders like cystic fibrosis and muscular dystrophy, which can be life-threatening. The less is more hypothesis suggests that the LOF variants at the genome-wide level falls into regions displaying signatures of positive selection, showing that the LOF variants have a selective advantage in human evolution. This means that instead of the loss-of-function variants being a loss, it actually is a gain. The less is nothing hypothesis occurs when the LOF variants and the chromosomes are tolerated because corresponding genes are highly redundant. This is seen in healthy individuals and does not impact the survival of an individual.
Figure A shows that disease-causing loss-of-function variants are not likely to affect heterozygotes. In the sample population tested, there were loss-of-function variants found on heterozygous genes. However, the genes that they were found on were non-essential. Figure B shows that none of the loss-of-function variants have a statistically significant association with complex diseases. This means that the probability of having a complex disease for a patient with loss-of-function variants is no greater than expected from random chance. By looking at these two figures, we can conclude that the less is nothing hypothesis was one of the hypotheses proven to be true during the experiment, showing that the loss-of-function variants do not affect the gene.
This stringent filtering of the 185 human genomes led to the identification of 26 known and 21 predicted severe disease-causing variants. However, this survey of loss-of-function variants also shows us that we need to be careful when assigning which gene-disrupting variants are disease-causing or not. This is because the scientists found that a typical healthy human genome contains approximately 100 loss-of-function variants, most of which are in the heterozygous state. Since human beings have been estimated to carry less than 5 lethal alleles per genome, it seems very likely that most of these loss-of-function variants are found in non-essential genes and are not disease-causing. Therefore, because most of these loss-of-function variants exist in the heterozygous state, Many alleles with large effects on human fitness exist at a low frequency in the human population.
The Human Genome Project and this experiment demonstrate a greater need for a more extensive comprehension of the human genome so that we are able to fully understand loss of function variants and their effects on human disease risk. If we continue to broaden our understanding of the human genome, we will obtain the ability to not only discover, but also to possibly prevent disease-causing genes that could have negative effects on human fitness. We will also be able to map the ancestry of certain genes and even pinpoint locations of gene prevalence throughout the different countries of the world. This experiment has great significance in the scientific society of today and shows us that we have a need for a greater understanding of our human genome.